We have been continuing in this lecture series over archegoniates. In our previous lecture, we have come across various aspects of biology of the genus Pinus. We have seen the distribution of Pinus, morphology of Pinus, and the anatomy of root and stem in Pinus. Our discussion of anatomy will not be complete if we do not talk about the secondary growth in Pinus. So as far as the secondary growth in Pinus is concerned, before the secondary growth begins or the secondary growth initiates, the fascicular cambium and interfascicular cambium join to form a complete ring. The cambium cuts off two types of cells, the fusiform and ray initials. The fusiform initials are elongated with tapering ends and are tangentially flattened. They give rise to the axial system, that is xylem and phloem. The ray initials are small and isodiametric and form the radial system, which is composed of rays. The cambium produces a continuous cylinder of secondary xylem towards the inside and secondary phloem towards the outside. The cambium also cuts off parenchyma cells, which produces secondary medullary rays, which are uniserrate and thick-walled. Now, as far as the tracheids of secondary xylem are concerned, they have bordered pits. The racine canals, they develop in secondary xylem. The secondary phloem has sieve elements, the most characteristic feature of which is the occurrence of sieve plate on the radial walls throughout its length. The sieve plates consist of numerous narrow channels lined with callos. The companion cells are absent, but albuminous cells seemingly execute the same function. Medullary rays are of two types, that is uniserrate rays and multiserrate or fusiform rays. Now, before we go and talk about different types of rays, let us try to understand some basic concepts about the planes in which the anatomy is being studied. So, let us consider this as a log of wood, and there is a longitudinal axis. Any section which is perpendicular to this longitudinal axis is also termed as transverse section or TS. So, this plane, the structures which are shown on this plane, they are representing the transverse sectional structures. Now, from the center towards the periphery, when we cut a cross section in the line of this longitudinal axis, such kind of sections are termed as radial longitudinal section. In short, we call them RLS. But when such longitudinal sections, they are cut away from the center, like here, such sections are termed as tangential longitudinal sections or TLS. In the second diagram, we can further elaborate these different types of planes. This is the core of the primary tissue, and in a radial longitudinal section, we see the xylem rays here and secondary xylem here, whereas in a tangential longitudinal sections, we see the xylem rays arranged over each other. Here in a radial longitudinal section, we see the wall of xylem rays, whereas in case of tangential longitudinal section, we see xylem rays as cells lying one above the other. So, once we have this concept of various planes in transverse section and or RLS or TLS, let us try to understand how these structures look like. So, when we talk about Pinus, the medullary rays in a radial longitudinal section, they show these distinct structures, whereas in case of tangential longitudinal section, they show these structures. Here in case of RLS, the medullary rays are seen here horizontally lying, whereas in case of TLS, they just look like a bundle of cells lying one above the other. The RLS depicts the length and height of the ray, whereas the TLS, that is tangential longitudinal section, show the height and width of the ray. The TLS through the xylem region displays xylem rays in addition to border pits. Xylem rays may be uniseriate or linear, multiseriate or fusiform. In the phloem, sieve tubes with sieve plates are present besides the phloem ray, which consists of starch cell in the middle and albuminous cell on either side. The wood structure of Pinus is much simpler as compared to the wood of dicots. The wood composes mainly of tracheids, and the characteristic feature is the absence of vessels. The cork cambium differentiates in the first or second layer of the cortex. It cuts off cork on outer side and secondary cortex on the inner side. Anatomically, the dwarf shoot appears like the long shoot except for its small diameter. Also, the secondary growth is limited in magnitude and the pith is large and parenchymatous. Now let us talk about Pinus leaf and its anatomy. In its anatomy, the Pinus leaf shows many xerophytic features, and it has adopted several modifications to withstand extreme conditions. The outline of the foliage leaf or the needle varies according to the number of needle in the dwarf shoot or the spur. For example, in case of Pinus monophylla, the solitary needle is circular in outline, whereas in case of Pinus mercussi, each spur bears 2 needles and outline is semicircular. In 3 needle pines, as the case is in Pinus roxburghi and Pinus wallaceiana, the outline is triangular. Here you can see the outline of a needle. So the outline of Pinus sylvestris and Pinus roxburghi. Since in case of Pinus sylvestris, single spur bears 2 needles, the outline happens to be semicircular, and in case of roxburghi, the outline is triangular, more or less triangular. The epidermis is single layer of isodiametric lignified cells coated with a thick deposition of cuticle. On this epidermis, numerous depressions are seen over the epidermis, in which the sunken stomata are very frequently found. The walls of the subsidiary cells and guard cells are partly lignified. The hypodermis composes of 2 or 3 layers of thick walled cells. Let us try to understand the outline of a leaf with the help of this diagram. Here the epidermis is very heavily cuticularized. This prevents any excess evaporation of water, and also it helps in protecting the leaf from direct sunlight. During winter, this cuticle layer helps in protecting the epidermal and hypodermal cell from freezing. So this epidermis is interrupted by multiple depressions. These depressions are nothing but sunken stomata. There are numerous sunken stomata which are found interrupting the epidermis, and below epidermis is found the hypodermis, which is more or less sclerenchymatous, and below this thick walled hypodermis are found these mesophyll cells are green and chlorophyll containing cells. They are primarily responsible for photosynthesis. Inside these mesophyll cells lies the vascular bundle, which has phloem and xylem, phloem towards outside and xylem towards the inside. On the 2 sides of the leaves, racine canals are found, one on each side of the cross section of the leaf. So as we have already discussed, the hypodermis is interfered by air spaces below each stoma. The dermal layer, that is EP and hypodermis, is followed by mesophyll surrounding the diarch or monoarch stele. The mesophyll is chlorenchymatous with varying number of plate-like or peg-like enfoldings of wall projecting into the cell cavity to increase the assimilatory surface. Mesophyll is not differentiated into spongy or pellucid parenchyma as we see in dicots. Racine canals also exist in the mesophyll, comprising of tubes lined with special cells that secrete resin. The endodermis is a single continuous layer of barrel-shaped cells. The pericycle is parenchymatous, multi-layered and interspersed with transfusion tracheids. A sclerenchymatous sheath encloses the vascular bundle. In case of Pinus roxburghi, there are two vascular bundles placed at an angle to each other. The xylem is made of protoxylem elements and metaxylem elements. Phloem composes of sieve cells and parenchyma. Now there is a unique set of cells or tissues which are found called as transfusion tissues, which occur on both sides of vascular bundle and consists of two types of cells, the tracheal cells and parenchyma cells. The tracheal cells are without protoplasm and bear pits. They aid in lateral conduction of water from the xylem to mesophyll. Whereas the parenchyma cells contain protoplasm and they are not pitted, they lie in between the mesophyll and the phloem, and they play a role in transport of food and nutrients. Now, as far as the anatomy of a young root or pinus is concerned, it mirrors to that of a dicotyledonous root, except for the absence of vessels in the xylem and companion cells in the phloem and the presence of racine canals opposite the protoxylem groups. The young long roots are circular in outline. The structure from outside inward is epidermis, followed by 4 to 5 layers of starch-filled cortex having an outer zone of small and inner zone of large parenchymatous cells. The innermost layer of cortex is single-layered endodermis with Casparian strip and is followed by 6 to 7 layered pericycle containing starch and tannins. In Pinus, both long and short roots are diarch or tetrarch. The primary xylem and primary phloem show radial arrangement. Each protoxylem point is linked with the racine duct. The metaxylem element are made up of pitted tracheids. The phloem alternates with xylem and consists of sieve cells and phloem parenchyma. The pith cells are rich in starch. Occasionally some of them are tanniferous also. The dwarf root of Pinus is anatomically similar to the long root. It, however, varies from the long root in lacking a root cap, racine ducts, starch in cortical cells and secondary growth. The cortex is similar in the dwarf root than in the long root. Now let us talk about the reproduction in case of Pinus. Reproduction in case of Pinus takes place by the production of spores which are born in sporangia. These sporangia are positioned on the sporophylls. The tree happens to be monoecious, that is the male and female cone are born on the same tree, but they are born on separate branches. The male cones arise on the tip of central and lower branches, and the female cones acquire a location on the upper branches for procuring an advantageous position for gaining the wind-borne pollen grains. Abnormal bisexual cones are also sometimes reported in Pinus maritima and Pinus montana. So let us talk about male cone and male gametophyte in a bit more detail. Each male cone is a cylindrical to ovoid structure about 1.25 centimeter to 1.9 centimeter long, yellowish green in color in juvenile stage, but they become reddish brown on maturity. They are usually produced in the spring. The male cones are formed in the axils of scale leaves at the base of a young shoot and replace the dwarf shoots. These male cone, they occur in clusters, and the number of male cones may vary from 15 to 140 in a cluster. There may be hundreds of such clusters on any single tree. Here in the diagram you can see there is a picture of a cluster of male cones. The number of male cone may vary from 40 to more than 100. Each male cone consists of a central axis on which numerous microsporophylls are arranged spirally. Microsporangia develop in pairs on the abaxial side of each microsporophyll. The cone axis elongates, exposing microsporangia which dehisce longitudinally. At this 4-cell stage of microspore, the microsporangia dehisce longitudinally and pollen grains are liberated. The 4-cell stage of microspore is because of the presence of 2 prothallal cells, 1 entheridial cell and 1 tube cell. In the microspores, a thin layer of callose first develops at 2 sites where wings are destined to be formed, and later spreads and covers the proximal end of the pollen grain. The pollen is now mature and prepared to pollinate the ovule. The uninucleate pollen grain is the first cell of male gametophyte. Each pollen grain is coated by a 3-layered wall. The outermost layer is called as exine. It is present only on one side of the microspore and is thickly cuticularized. The middle layer or the exoentine covers the rest of the spore and forms balloon-like projections or air sacs, also called as wings. These wings furnish buoyancy to the spore and aid in its dispersal by wind. The innermost layer, intine, is very, very thin. You can understand these structures with the help of these pictures and diagrams, where there is a longitudinal section of male cone. On the central axis, the microsporophylls are arranged spirally, and in longitudinal cells they are shown on opposite sides. Now each microsporangia, they have numerous microspores. These microspores, they metamorphose into 4-celled pollen grains. Each pollen grain has 2 prothallal cell, 1 entheridial cell and 1 tube cell. Upon reaching to the female gametophyte, the tube cell gives rise to a long tube, and thus the fertilization process will start taking place. Here you can see the formation of a male gametophyte of Pinus. From a single cell, the mitosis takes place, followed by the formation of 4 different cells. These 4 different cells, 2 of them represent the prothallal cell, 1 is the central cell and 1 is represented by the tube cell. Here you can see the formation of tube cell. Now let us talk about the female cone or the female gametophyte. The female cones are much larger than the male cones. The female cones replace the long shoots, unlike the male cone which replace the dwarf shoot, and the number is variable. They are normally solo, but maybe 2 or even in 1 or 2 whorls of 3 each are found in one cluster. The female cone takes slightly more than 2 years to complete maturation and seed formation. At first, the cones appear red or purple in color, then turn green in 2 seasons and finally mature into brown woody structures. Each female cone has a central axis on which 80 to 90 megasporophylls are arranged in a spiral fashion. These megasporophylls are of 2 types, the large bract scales and the smaller ovuliferous scales. The large bract scales are arranged spirally and develop directly from cone axis, while the ovuliferous scale, they develop on the upper surface of the bract scale. The bract scale is leathery while ovuliferous scale is woody. The bract scales are hidden under the ovuliferous scales and therefore are not visible from outside. The bract and the ovuliferous scales together form a seed scale complex. Each megasporophyll bear 2 ovules on the upper side, or the also called as the adaxial surface of these megasporophylls. The ovules are inverted with micropyle facing the axis of the cone. The ovuliferous scale is initially much smaller than the bract scale, facilitating the entry of pollen grain. But in post-pollination stage, it overgrows and later ending in blocking the space between the bracts, and thus the cones get closed. Each ovule of Pinus is covered by a thick layered integument. The integument is fused to the nucellus except near the micropylar region. One of the integument layers later turn into seed coat. This can be seen with the help of these diagrams. These 3 cones represent the developmental stage of female cone of 3 ages. So this is a female cone of 1 year old, this is a 2-year-old female cone, and this is the 3-year female cone and a mature one. In a cross section, this mature female cone looks like having a central axis, and on the lateral side they have a megasporophyll. On the megasporophyll, on the adaxial surface, they have 2 ovules. These ovules, they are inverted and they have a micropyle, and the entry of pollen tube takes place from this. Now, over a period of months, the functional megaspore increases in size considerably. Its nucleus also enlarges and undergoes several free nuclear divisions, leading to the formation of numerous free nuclei to give rise to the free nuclear gametophyte. The number of nuclei in the free nuclear gametophyte is fixed for a particular species. After free nuclear divisions, the large vacuole appears in the center and all nuclei get arranged near periphery. Later on, wall formation starts centripetally. The first walls are laid perpendicular to the megaspore membrane and gradually proceeds to the middle of the central vacuole. These cells are like long tubes that are open towards the inside and are called alveoli. Afterwards, they become closed and new cross walls are laid down in these tubes. They form the endosperm or female prothallus, also called as the female gametophyte. Each archegonium has its own archegonial chamber. Cells surrounding the archegonium form the jacket layer, which supplies food to the developing egg. Numerous pits are present in the inner thickened wall facing archegonium. The archegonium keeps its contact with the surrounding gametophytic tissue through these pits. When the pit membrane breaks down, the various organelles and even the whole nuclei moves from the jacket to the egg cytoplasm. So finally the fertilization and pollination takes place, and since the Pinus is wing pollinated, at the time of pollination the yellow colored pollen are produced in such great quantity that the hill slopes in a pine forest appear to be painted with these pollen grains, which emerge like sulfur dust and hence called sulfur shower or sulfur rain. At this time, the female cone is still open and wind pollen grains easily pour over, roll down the obliquely tilted scales and finally come to the rest in the vicinity of micropyle. At this stage, the ovule secretes a drop of sugary liquid called as pollination drop, which fills the micropylar cavity and exudes out at the flare tip of the integument. So with the help of wings, these pollens are oriented in a manner that the germinal pore of the pollen contacts the drop and is sucked inside the ovule. The pollen tube arises from the intine. While moving through the nucleus, the pollen tube discharges enzyme disintegrating the nuclear cell walls. Thus, the fertilization will take place. So, this was all about the reproductive structures, the methods, the process of reproduction, and also the fertilization. And thus the entire life cycle of Pinus gets completed. I hope I have been able to cover up almost every biological aspect of the life cycle of Pinus. Thank you so much for your patient listening.